Hello everyone, I am excited to share our work on emotion processing in brain in the individuals with misophonia and hyperacusis, two increasingly recognized sound tolerance disorders. Imagine sound sensitivity as a volume knob. Most people have a healthy upper limit. Loud sounds are noticeable and tolerable. But for individuals with hyperacusis, that maximum is set too low. Everyday sounds become unbearably and painfully loud. In misophonia, it is not loudness sensitivity, but sensitivity to very specific sounds in the environment, such as chewing, whistling, slurping, tapping, which trigger intense emotional reactions. These two disorders fall under the same broader category of decreased sound tolerance disorders. Despite this, these two disorders have rarely been studied side by side. We do not yet know the unique neural signatures of each of these disorders and when they co-occur. So the aim of our study was to examine behavioral differences in emotion perception across hyperacusis, misophonia, comorbid when both conditions co-occur, and control groups. And the second aim was to investigate neural differences in the engagement of the emotional processing network in individuals with sound tolerance disorders and without. We recruited a young collegiate population aged between 18 to 25. All participants completed hearing hyperacusis and misophonia questionnaire. They were invited in lab to complete detailed hearing assessments, and structured clinical interviews were conducted by a clinical psychologist. Then the participants were invited to complete a task-based functional magnetic resonance imaging study. In this paradigm, we presented 90 emotional sounds while the participants were in the scanner. These emotional sounds were extracted from the International Affective Digitized Sounds database, where the sounds were categorized as pleasant, neutral, or unpleasant. The participants were instructed to provide perceptual rating as soon as they felt confident in their rating. Participants were categorized into 4 groups based on their hyperacusis and misophonia questionnaire scores, and their demographic and questionnaire scores are given here in the table. Based on their hearing assessments, all participants had normal hearing thresholds. Moving on to data analysis, the acquired fMRI data was pre-processed using standardized pre-processing steps like head motion correction, normalized to a standardized space, and smoothed. We conducted first level analysis and created contrasts of interest, that is unpleasant greater than neutral and pleasant greater than neutral contrast. At second level, we conducted one-way ANOVAs. We also conducted a region of interest analysis using multivariate ANOVA. We identified regions of interest based on previous literature, and the 8 ROIs are given here on the slide. We will first look at the behavioral results. We collected participants' reaction times, that is the time it took them to categorize sounds in 3 different valence categories. The top plot here represents condition on the x-axis and reaction times on the y-axis. We did not find between group differences, but we did find a significant main effect of condition, where all groups reacted faster to unpleasant and pleasant sounds compared to neutral. Now we turn our focus to the bottom plot here, which represents condition on the x-axis and perceptual ratings, that is the number of sounds categorized in each of these categories on the y-axis. Here we found a significant interaction effect of group condition, where the comorbid, that is misophonia hyperacusis group, rated more sounds as unpleasant compared to the misophonia and control groups. Similarly, on pleasant sounds, we found that the comorbid group rated fewer sounds as pleasant. It is important to note that we presented 30 sounds across 3 categories, but fewer sounds were rated as pleasant across all groups. Now we look at the neuroimaging results. We conducted whole brain analysis using one-way ANOVA and only focused on the unpleasant greater than neutral contrast. We did not include the pleasant trial as it had fewer trials. On the one-way ANOVA, we found a significant effective group which was localized in secondary visual areas and association cortices in the left hemisphere. Here, this box plot here represents group in the x-axis and contrast estimates on the y-axis. And we find a significant effective group, and the group with comorbid condition exhibits greater activation in the left secondary visual and association area compared to the control group. Now we turn our focus to the region of interest analysis, where we consider both unpleasant greater than neutral and pleasant greater than neutral contrast, as this is a much more focused analysis. We found a significant main effective group on MANOVA. Now we look into each of the regions of contrast. We found significant group differences on bilateral primary auditory cortices, which are represented here on the left plots. Misophonia group shows greater activation compared to the hyperacusis group. We also find between group differences in anterior insula, where hyperacusis group shows reduced activation compared to the control group. We do not find between group differences on anterior cingulate cortex and the orofacial motor cortex. Now, looking at the key findings, the comorbid group rated more sounds as unpleasant, indicating altered affective sound perception in the comorbid group. The same group also showed enhanced visual cortex activation, indicating increased multisensory attention or hypervigilance for aversive sound processing. We also found that the hyperacusis group showed diminished neural activation in key auditory and salience regions compared to misophonia. These preliminary findings indicate distinct neural signatures for misophonia, hyperacusis, and their comorbid condition, and take us one step further to understanding these conditions. Thank you for your